Hello, everyone, and welcome to this very exciting lunch and learn session. So, I'm Kenichi Okuda, UNC Chapel Hill. It is truly great pleasure to co-chair this exciting lunch session with Dr. Viral Sharaf, Harvard Medical School. So this session is focused on very important topic in the CFTR molecular therapy. So we are gonna discuss, of course, like having a lunch, but we're gonna learn and discuss which cell types are ideal therapeutic target in the CFTR molecular therapy. So before talks from our three expert invited speakers, I'm gonna just really briefly go overview the current paradigm in this topic. So to understand which cell types are a therapeutic target for the CFTR molecular therapy, it's very important to know which cell types physiologically express CFTR. So in the early days back in 1990s, Dr. Engelhardt's group, he's also the invited speaker this session, identified CFTR expression in non-ciliated cells in the human bronchiole, and also very interestingly pointed out CFTR-rich cells in submucosal ductal epithelium. Later on, Dr. Kredat's group identified the CFTR expression in the apical membranes of ciliated cells, but also pointed out CFTR-enriched hot cells in human large airways. And after that, more recently, Plaschard and Montrose group identified finally pulmonary ionocytes, which are rare, but express substantially higher levels of CFTR. This is data from Human Lung Cell Atlas showing CFTR expression in the variety of epithelial cell types. So this data again highlights very exclusive feature of ionocytes expressing high levels of CFTR. And these the percentage on the top of each bar shows how many proportion of CFTR positive cells in each cluster. So ionocyte, 80% of ionocyte annotated cells express CFTR. But this data also highlight relatively lower level, but CFTR is also expressing other cell types, including secretory cells like club, goblet cells up to 16 or 80% per each cluster, and fewer and then less cells in the basal and then ciliated cells. So, and these data raise several key questions. For example, which cell types normally express CFTR, and which cell types are most relevant, functionally relevant to CF pathogenesis, and perhaps does really targeting specific cell types matter for CFTR therapy? So, I mean, we should target progenitor cells like basal cell to eventually correct CFTR in all these cell types. So to address these questions, now the field is moving from identifying cell type specific CFTR expression to identifying cell type specific CFTR function. So our expert speaker is there is already very exciting discussion this morning's session, but our expert speakers update the latest data set for cell type specific CFTR function.
Great. As Kenichi mentioned, there's a lot of important questions. Fortunately, we have people who are going to answer all of our questions about what cell types to express CFTR in. And so we have three wonderful speakers here, Dr. John Engelhardt from the University of Alabama, Dr. David Stoltz from the University of Iowa, and Dr. Juan Ionocytes from University of Saskatchewan. And so, like I said, we'll do 15-minute talks, 10-minute discussion, but hopefully we can keep this a little bit more informal. We all have lunch here. There's no alcohol, unfortunately, but maybe that'll, also help some discussion. But before further ado, Dr. John Engelhardt.
All right. Well, thanks, Viral. Thanks, Kenichi, for the invitation. I'm going to talk about a subject that's near and dear to my heart, and I've tried to spend my career getting answers. No disclosures related to the current talk. So as you heard, there are a number of different cell types within the airway, of which a small subset express CFTR in about 15 to 80, 90% of cells when one considers ionocytes. Yeah, that'd be helpful to set that, or I'll go forever. As I mentioned, about 1% of the epithelium is in the proximal cartilaginous airways composed of ionocytes. but they express about 50 to 70% of the CFTR mRNA in this region. So we've used ferrets to study this, mainly because the cell biology of their lungs and the distribution of the submucosal glands are very similar to human. In mouse, submucosal glands are limited to the very proximal trachea. And we generated a number of different transgenic models that I'm going to be talking about, including a Cree reporter, FOXI1 Cree. FOXI1 is required for specification of ionocytes, so it marks them very specifically, and conditional knockout at CFTR locus. And so if we use these top two, and we label with tamoxifen to induce at the estrogen receptor on Cree, one can mark ionocytes, both in the glands and in the surface airway epithelium. And if we included this conditional background here, these ionocytes would lack CFTR, and I'll show that. Then we recently created and published in a paper by Fang Wang a conditional knock-in model. This is an exon trap in which EGFP and the endogenous CFTR3' UTR is knocked into the first intron of CFTR and captures splicing followed by 3 stops. At homozygosity, it's essentially a knockout. And so simplistically, the approaches we were trying to use to understand CFTR function on ionocytes are two. One is to specifically knock out CFTR and ionocytes in a wild-type background, and the other is to specifically knock in CFTR, much like you might do with gene editing, only in ionocytes in a cystic fibrosis background. So this is just an example in which you can see knocking out CFTR with this approach. These BSND expressing ionocytes all have these apical caps of CFTR expression. You tamoxifen treat, this is whole mount trachea, and one loses CFTR protein. And conceptually, the reverse is this knock-in model where EGFP is expressed in BSND expressing ionocytes that don't express CFTR at homozygosity, and if one induces with tamoxifen, you lose EGFP and you induce CFTR. So this is some electrophysiologic data showing chloride absorption in air-liquid interface cultures from conditional knockout, ALI proximal airway, starting in symmetrical chloride, shift to low chloride. And here is the CFTR expression, where with the low chloride gradient or CFTR function, you can have CFTR as well as other channels contribute to this. Forskolin IBMX induces CFTR and GlyH101 inhibits. And if you compare this to this is vehicle treated. You compare it to hydroxytamoxifen treated, which induces CFTR excision, completely lose this current, this absorptive chloride current, and it's equivalent to G551D in the absence of modulators, which is CF. And you can quantify this, and it's essentially a complete loss of CFTR activity equivalent to whole cell knockout in all cell types. So our interpretation is that CFTR is required for the ability of pulmonary ionocytes to absorb chloride and thus also absorb fluid. And I'm not going to talk about those fluid absorption measurements because they're published. Now, the reverse of this is to now invoke a low symmetrical chloride on both sides, and on the basolateral side induce high chloride. So now we're going to drive secretion and then ask is CFTR and ionocytes required for that. And here's the wild type vehicle treated conditional knockout cells. And when you induce CFTR deletion within ionocytes, one sees a decrease in all 3 phases. And basically the area under the curve here would be the CFTR dependent change in current in chloride secretion. And CF is nearly completely ablated here. So this residual current here is likely due to secretory cell expressing CFTR. And this inhibition is about 3-fold to 2-fold, depending on whether or not you're looking at forskolin induction or GlyH101 inhibition of the channel. So CFTR in our hands is required for pulmonary ionocytes to secrete chloride and thus may participate in fluid secretion. But ultimately, there's been some debate about whether or not short-circuit current can be used to evaluate the ability of pulmonary ionocytes to secrete fluid. And really, ultimately, we felt that using a organoid model, which we have here, conditional knockout with vehicle, you can see swelling And if one knocks out, this is with vehicle, one knocks out what's hydroxytamoxifen, so these cells this organoid doesn't have CFTR and ionocytes, one sees it's not swelling. And you can quantify this as the integrated area under the curve, and there's really a significant difference between vehicle treated down here and CFTR knockout, or CFTR vehicle treated here and the knockout down here. And one can quantify this, and there's some very interesting aspects about this. We did add amiloride to this to inhibit competing absorption and secretion, but in the absence of amiloride, it's maybe only about 20-fold reduced secretion. But the shrinking of these organoids here that lack CFTR induced with amiloride and forskolin suggests that there are other channels other than ENaC and CFTR that can control secretion within the organoid. So CFTR expression in pulmonary ionocytes is required for fluid secretion under open circuit conditions and following pKa stimulation. And as I mentioned, the amiloride only enhanced secretion about 20-fold under these conditions. So is CFTR and ionocytes required for mucociliary clearance? These studies were published in Nature, but we used a gadolinium PET-CT imaging to look in the proximal airways deposited in the very distal trachea and bronchi, moving up to the larynx up here. This is baseline mucociliary clearance in a FOXO and Cree conditional CFTR knockout. So these animals were imaged first, then induced, and a week later re-imaged, and a significant difference in clearance. So CFTR expression in pulmonary ionocytes is required for effective MCC in our hands and in the ferret. Now, the flip side of these experiments, the conditionally knocking out on a wild-type background, is to have a gain of function on a cystic fibrosis background. As I mentioned, this is sort of the theoretical gene editing experiment where 100% or close to it ionocytes on a CF background regain CFTR function. So what are the results from this experiment? And I'll just point out that we had to make these compound heterozygotes because we needed to rear the animals and keep them alive. So after they were born on CFTR modulator, they have one G551D allele and then the knock-in allele on the other. And so here's an experiment where we're looking at chloride secretions. So there's a shift to low apical chloride at this point. The bars in blue are the ones that are recovered for CFTR, so hydroxytamoxifen treated versus the vehicle. And you can see that significant difference in the amount of current, secretory chloride current that's regained by reactivating CFTR and ionocytes. And the flip here is chloride absorption. And you can see in the absence of CFTR and ionocytes, there's no chloride absorption following induction. And this is the gain, the relatively significant amount of current here for only heterozygosity of CFTR. So restoration of about 50% of CFTR and ionocytes can restore a very large portion of the chloride secretory and absorptive currents. So what's going on here? Well, we propose that ionocytes can, in a cell-autonomous way, both absorb and secrete chloride and drive fluid absorption or secretion. And that doesn't mean that there isn't communication with other cells around them that's participating in this, but it means that they can function semi-independently. And I didn't talk about things that were published in this Nature paper that show that ionocytes are also required for pH regulation, volume regulation, so they dehydrate in the absence of ionocytes, and mucus viscosity and clearance. So this is the big question, are ionocytes required targets for gene therapy? I spent my career thinking, well, probably they are going to be. And to rush the punchline, that may not be the case. So this is a study that we collaborated with Arcturus using an LMP to deliver, in this particular instance, deliver Cree into these Cree reporter Rosa TG ferrets. You can see very high level transduction in the conducting airways as well as in the surface trachea. There's about 40% in the interlobar airways and 40% transfection within the trachea to convert the Cree reporter. So obvious question, what cell types are transduced? Well, predominantly here stained for FOXJ1, which is a ciliated nuclear cell marker. You can just look at this, and this membrane-bound GFP tracks to the cilia of airways. So that's why you see these caps that are really hot, but that means it's a ciliated cell too. So most everything here is ciliated cell transfection. There, you look down here for a mucus marker, and most goblet cells are not positive for GFP. Here's an example of one that I think obviously has some GFP in the cytoplasm overlap with the mucin, and this even looks like it's potentially a ciliated goblet cell, which do exist in disease states, by the way. So staining for BSND, we could see evidence of transfection of ionocytes, but this was fairly infrequent within the samples that we evaluated. We didn't quantify the ionocyte transfection because they're so infrequent, it's very difficult to do that with the numbers we had, but here is quantification of ciliated cell transfection, about 40%, 30-40%, depending on interlobar airway or trachea. And the axis here, I'll say, is different. So this is about 1.5% on average for secretory cell transfection. If one looks at this as tomato mRNA expression, it's just another way to look at the level of transduction at these levels. I'm not going to show you the cell marker staining, but mostly also ciliated cell. But here's CFTR staining. You can see CFTR in the apical boundary here, right below this cilia. This is the vehicle-treated control. You can see here, probably this is an ionocyte with very definitive cap, and maybe this is a high-level expressing secretory cell. So can lunar mRNA complexes correct mucociliary clearance defects? This is the big question. And so we performed a study in G551D animals, and I'm going to try to convince you that this clearance is an index of CFTR function using G551D ferrets and ivacaftor VX770 to modulate CFTR activity. And so we took these animals and waited until they had a range of clearance that was about 20 to 40% of wild type, and then did a study with human CFTR delivery mRNA or tomato as a negative control. This is the experiment to convince you that this assay detects CFTR activity. We're looking at clearance here from the distal trachea up to the proximal trachea. These are ferrets on VX770, fairly rapid clearance. These are ferrets off VX770, so CF ferrets in this latter two group. And if one quantifies this as the percent cleared, you can see baseline starts up here in blue, and over time of removal of VX770, you lose clearance. You put the animals back on. 2 weeks later, it's back up to wild type. And you can quantify this as the clearance rate here at the initial rate that's linear. And you can see this trend over 4 weeks. Predominantly, they lose most of the mucociliary clearance and can be regained. So we believe that this assay reports CFTR activity in mucociliary clearance. So now we're comparing both tomato transfected as well as human CFTR. Here is baseline studies in the animals that used to classify them at 20 to 40%, then delivered the lunar CFTR, human CFTR mRNA, and 24 hours measured clearance. And you can see a significant increase. Here are the images. And if one looks at the tomato, there's really no change in the extent of clearance between the tomato and the baseline studies. One can quantify this again, and here just the clearance rates, very significant differences between these groups and recovery in the human CFTR. And in nearly every ferret, there was an increase in clearance with human CFTR and lunar, and really no tangible increase in the tomato mRNA transfected cultures. So in summary, we believe that CF and other transgenic ferret models can aid in determining what cell types, what's the pathophysiology for CFTR expression in various cell types, what are potential therapies. The LMP lunar transfects primarily ciliated cells, yet it improves mucociliary clearance very close to wild type levels. And while I've spent my career studying specificity of CFTR at the cellular level and what its functions and implications might be for gene therapy, I'm actually encouraged for the field that we may not need to reconstitute CFTR in ionocytes and the 30% of secretory cells that express tenfold lower levels of CFTR. That's a really good thing. Physiologically, how can that happen in an animal? Well, will take another 5 years to probably figure out for sure. But, there are a lot of things you could hypothesize about, that cells react to changes in membrane potential by regulating other channels to reach equilibrium. We know this because if we knock out CFTR and ionocyte, they change channels within those ionocytes that now lack CFTR. If the same thing happens in a ciliated cell, then maybe a ciliated cell can get partway to becoming ionocyte or performing secretory cell functions, potentially moving bicarbonate. So I'd like to thank a lot of people in the group and collaborators that have done this. Feng Wan, Java, Grace Grazer did a lot of the studies on ionocytes, and Xiaoming Liu and his team on the Arcturus, evaluating the Arcturus LNPs. Thank you.
Great. Thanks so much, John. Let's have some questions from the audience. Dr.
Going to have some discussion, right? So a couple of things. So in the rescue experiments, did you transduce the submucosal glands? Do you know?
With that particular LNP, not much. I mean, maybe a little bit in the gland ducts that have access, if the LNP gets in there, but not deep within the glands where CFTR is expressed, like in serous cells.
And so do you rescue the mucociliary clearance because you've promoted secretion or absorption?
Or a combination of both. How can you not correct the submucosal gland defect and still correct mucociliary clearance? It could be disease stage dependent. When the studies that I showed you in the CFTR, in the G551D where you move VX770, those are about a month off. That's really early stage disease. If you get out at 5-6 months off with a big mucus burden and where glands may be contributing more to defects in the clearance, the story could be different.
Well, so this relates to my final question. When you talk about absorption and secretion and you're using an Ussing chamber with a chloride gradient one way or another, it's CFTR dependent, the chloride currents that you're measuring. And we saw this morning that CFTR is expressed in the apical membrane. You're measuring a transcellular chloride current either in the absorptive direction or in the secretory direction. What is the pathway for chloride in the basolateral membrane? It's not CFTR. Is it Barton? Is that the...
So there is CLC-NKA is enriched in B, and BSND is part of the complex for absorption. If you're asking how can the same cell control both absorption and secretion, I'm not sure that that's actually the case.
I would suggest that the use of those two terms in asymmetric chloride gradients is incorrect. You have the open circuit data in the organoid shows secretion.
In the Nature paper, I didn't show the data where we looked at over time fluid absorption and secretion by measuring ASL height in ALI cultures that didn't have no ionocytes. And what you see is that the absorption is slower. And then once you hit this critical dehydrated state in a wild type epithelium, you activate secretion. And it's almost a sinusoidal equilibrium of that height. That second phase of secretion is lost when you don't have ionocytes. So lack of ionocytes dehydrates the airway surface fluid. That's under open circuit conditions. It's not. And then the other data I didn't show is movement of chloride specifically through ionocytes. That's in that paper too.
Thanks.
But I appreciate what you're saying. But we use the gradient mainly to compare interpretations across the field of interpreting chloride secretion and absorption.
Very nice, John. I wonder when you sleep. But anyway, I think we can learn something from the modulator experience, right? Two things. First of all, as we discussed a little bit earlier, the modulators go everywhere, right? So no one worried at Vertex whether they're hitting the right cell type or not. They gave a small molecule that was biodiffusible and tried to hit every cell they possibly could, and assumed they did. The second point is, the other thing we learned from modulators is, and Jeff Wine just left because he was one of the first people to propose this, recovery of a small amount of CFTR function has a disproportionate effect on phenotype. And we'll present additional data from CFTR2 as well as from primary cell studies that support the idea that there really is a logarithmic response in recovery of function and improvement in of CFTR function, and improvement in lung function and drop in sweat chloride. So you showed evidence that you had a small number of ionocytes per hat which were corrected. Is that sufficient? Maybe correction of a small fraction of the ionocytes and a small fraction of these other cells is sufficient. Because once you get wild-type CFTR functioning, you've opened the dam and things start returning to normal again. So are we asking too much of what we're trying to do with gene therapy?
So narrow in on your question. So,
Be very specific.
I'll comment about a couple of...
Well, I went. My point with modulators is recovery of a small amount of function, okay? Even in certain genotypes gets you quite a big, in CFTR function, gets you a pretty big bump in lung function, right? So if you are just recovering function in a subset of ionocytes or a subset of secretory cells or a subset, is that sufficient recovery of function to get over the threshold to improve the phenotype?
Yeah, it's a good point. Comparing to modulators, a small amount in all cells that express CFTR, so moving up to 12.5%, which is something you've quoted in the field from splice mutations of being necessary to be at least have some activity that you don't get CF. It's a different scenario than targeting one. The experiments I showed were theoretical. It's basically one cell trying to figure out what is that cell doing from the standpoint of ion movement and fluid movement. So now if we could induce and maybe 10%, 20% of the ionocytes flip the switch of activating CFTR and do that sort of a study of what percentage of normal do you get back. I think it's an interesting question because, all these channels that regulate fluid transport in the absence and presence of CFTR, they regulate each other. To maintain cell homeostasis, at least I believe that. So if you have a cell that's heterozygote for CFTR and it's sensing that its environment of the airway surface fluid is defective, it's going to upregulate CFTR as best it can. And we know this even happens with ionocytes. And Fong, I'm not sure, I had to leave his talk early. Wang, I'm not sure if he showed when you actually turn off CFTR in ionocyte, the basal cells sense and specify new ionocytes that have wild-type CFTR in them, and you get recovery of current. So, I look much more now at the epithelium as being this integrated, very complex tissue, where cells are sensing what the other cells are doing or not doing right, and then as best they can trying to adjust it through other channels or specifying more cell types. And it's in disease when that goes awry that you really get, disease.
Yeah, and the point I'd make is, what is the lower threshold of your detection of correction in these situations? Maybe you're getting 5% correction in some of the cells. You can't tell, but that makes a difference.
Well, we can specifically ask what's the lineage trace efficiency. Now, we haven't isolated the ionocytes from, and asked at the single cell level, are both alleles, have they both flipped and activated? But by Western blot, looking at the protein level, it looks like, you're getting close back to.
But you can adjust the dose of tomography.
We could adjust tomography.
Do that and
dose it. And it's an interesting idea. I like that.
Oh, hey, John.
Yeah. Oh, yeah.
I'm just so amazed at the things you can do with ferrets. It's just unbelievable.
Thank you.
So one simple technical question in the swelling assays, have you looked at the cell types that are present in those organoids? Because if there's this debate, ionocyte versus secretory, they both have to be there for that to be a valid model.
Yeah, we haven't quantified that. The ionocytes are there, ciliated cells are there, the lumen, the cilia are on the inside. That was a reviewer question that we had to get for the paper in the Blue Journal. And so, we've studied a lot in organoids. They have secretory cells, they have basal cells on them, but we haven't quantified it for these specific experiments.
Well, the less easy, maybe non-technical question is what's happening in the distal airways in those ferrets, especially in the LNP, delivery studies.
Yeah, how does that contribute? Super great question. I think I had on there distal airway is different on one of my slides. If it wasn't, it should have been, because at least it's thought there are less ionocytes, or maybe the ionocyte-like cell that's down there is more like a club cell related to that. Who knows?
Yeah, I guess the real question about the ferret is like, what is the extent of distal lung disease in the knockout ferret, right? So it's not, I mean, the defect is there in the trachea, but is the defect there in the distal airways with the ionocyte knockout?
Oh, with the CFTR specific
Yes.
knockout. Okay. But that's also impacts my thinking of the progression of disease. So in the ionocyte knockout, we haven't looked in the like aged animals old enough in the conditional CFTR knockout in ionocytes. And I don't think it's actually possible to do the study because you knock out CFTR and more ionocytes specify that have CFTR, and you can't keep an animal on tamoxifen forever. So unfortunately, I don't think I can do that. But FOXI1 knockout, those animals, when you age them, granted, it's only like 5% of the animals, you can get to close to adulthood. So it's a selected bunch that survived. They don't get classic CF. I mean, we see all the defects in the epithelium and the proximal airway. I mean, the dehydration, the absorption, but they're not getting mucus plugging, at least in their lifespan, which is relatively short. But so that's made me think, maybe distal airway is where the disease begins, because the proximal airway has the phenotype of CF, at least in vitro in ALI cultures.
Hey, John, great talk. I was wondering what the voltage is across an organoid. Do we know that?
We haven't punctured, we put a probe inside
Right.
and outside the organoid.
But if the voltage is sufficiently depolarized compared to native airways, the organoids would secrete, the ionocytes in the organoids would secrete.
Well, how would they if it was, so you mean if the resistance was so low?
Yeah, and amiloride works from the basolateral side, so I mean some big molecules can get inside the organoid.
I guess I don't understand the question. I mean, Rick's not here, is he? Yeah. So, I mean, we haven't measured the resistance or the voltage across the organoid. If the organoid was, you mean if it was so loosely connected, I don't know how it would maintain swelling. I mean, it has to be at least tight enough. Now, maybe they have altered.
I mean, a small change in conductance can get rid of some of the voltage. It doesn't have to be a giant hole.
Go ahead, Kevin.
The water's only going to go where the salt goes, right? So the salt must be going into the lumen, the water is following.
Right.
Independent of the, there is a permeability barrier. It's swelling, it's not collapsing.
Yep.
So, I don't understand your question.
Well, so amiloride has to act inside the organoid.
Sorry.
Amiloride has to act. The site of action is inside the organoid, and so the amiloride in the experiments has to be crossing into the lumen, suggesting that.
Oh yeah, we had that question as a reviewer.
It's very self-permeable.
Yeah.
Yeah. No, I mean, it'll just, even an intact epithelium, it'll go from the basal to the apical.
But if you put it on a basolateral side of an oozing chamber, it does nothing. I mean, to airway.
Yeah, I don't know. People have used amiloride in organoids. We had that same question, a reviewer question.
Yeah.
How does it get in there to inhibit?
Yeah.
But there is a delta. I mean, if you do it with and without. But
No, I completely agree. It's just it's interesting that in an oozing chamber, right? It doesn't get to the other side for an airway culture, but in an organoid it can. Maybe the voltage is low.
Yeah, I mean, I'd be the first to admit, I think that the polarized ALI cultures are probably better differentiated from the standpoint of actually seeing the pseudostratified epithelium with higher epithelial height than you would typically see in an organoid. But
Yeah. Thanks, John.
John, I have one quick question I just want to jump on here. So you're probably thinking about this, but have you done an experiment with, do you have a FOXJ1 Cree-ER ferret that you could rescue CFTR in ciliated cells in the ferret model and then take a look at that? And then would they upregulate NKCC or something like that at that
I think
point?
that's a super great question. We haven't pulled the trigger on making a FOXJ1 Cree driver, but I'd like to do it for a lot of different reasons, basic cell biology reasons. But it's a really super good experiment and it's on the docket, but haven't pulled the trigger yet.
Gotcha. Well, let's thank John, everyone. All right. Let me just reset this for you, Stalls. Excellent. Next, we have Dr. David Stoltz from the University of Iowa.
Thanks, Viral and Kenichi, for the invitation to present this morning. Two quick disclosures. One is I'm not going to tell you which cell type is important. And second, you're not going to hear anything about pigs. So Viral and Kenichi asked me to talk about some of our recent published work looking at the effect of IL-13 on pulmonary ionocyte number and function. Most of this work was led by Guillermo Romano Ibarra, Ian Thornell, and Lei Lei. So essentially, these are going to be my take-home points. The first is I'm going to focus on the role of CFTR-rich ionocytes and how they mediate airway liquid absorption. Second, I'm going to show you data that IL-13 simultaneously decreases the number of CFTR-rich ionocytes, but increases the number of CFTR-rich secretory cells, ultimately increasing airway surface liquid volume. I'm going to show you an example in human disease, specifically asthma, how ionocyte number and function are altered. And finally, I think really the most important question is we clearly still have a lot to learn about how CFTR is regulated in the ionocytes as well as other channels. So a little bit of background I'll start out with in terms of what is the function of pulmonary ionocytes, and this is work of Ian Thornell that's been published. And so Ian was specifically interested in looking at what's the role of an ionocyte in human airway epithelia. And so he used cultures at the airway liquid interface, and he studied control airway epithelia and then airway epithelia where he increased the number of ionocytes by overexpressing FOXI1. First looked at electrophysiology, and so here I'm showing you delta short circuit current, CFTR mediated chloride current, and as expected, under voltage clamp conditions, you can see that increasing the number of ionocytes increases the amount of CFTR mediated chloride transport. So that was pretty much expected. Which was unexpected is that when he took these airway epithelial cell cultures, control and with increased number of ionocytes, and looked at them under non-voltage clamp conditions, you can see that the ASL volume was significantly reduced when he had more ionocytes present in the airway epithelia compared to control conditions. This suggested that ionocytes were increasing liquid absorption. And so he looked at fluid flux, and essentially that's what he found, that when you had an increased number of ionocytes, you had increased absorptive rates. He did the converse, so essentially knocking out ionocytes by targeting BSND, which encodes the Barton subunit of the chloride channel, and he found the opposite. So when you decrease the functional capacity of these ionocytes, you see increased airway surface liquid volume and decreased absorptive rates. So these were data are consistent with pulmonary ionocytes mediating liquid absorption. So ionocytes, chloride absorption, liquid absorption, secretory cells appear to be, at least in this model, predominantly chloride secretory and liquid secretion. And it's the ionocyte-specific Barton chloride channels in the basolateral membrane that are key for this absorption. So here's a schematic of what's probably happening in a healthy airway epithelia. But what we were interested in is what might happen in disease states. And specifically, we looked at what might happen under Th2 type disease processes, specifically asthma and COPD, where you typically have a lot of interleukin-13, which we know causes goblet cell metaplasia. And under these conditions, you have many more goblet cells, increased mucus production, increased chloride secretion, leading to increased airway surface liquid volume. But nobody had really looked at what is happening to the ionocyte under these conditions. And so we asked what happens to the ionocyte population with goblet cell metaplasia using IL-13 exposure. So we took human airway epithelial cells, cultured them in the presence or absence of IL-13, and then did a number of different assays. So first, we just confirmed that IL-13 in our hands would cause goblet cell metaplasia. So here I'm showing you low power images top down of human airway epithelial cells cultured at the air liquid interface and goblet cells stained with MUC5AC. And you can see really not many goblet cells present under these conditions. We quantified this. So here on the y-axis, I'm showing you percent of goblet cells in the airway epithelial culture over time with IL-13 exposure. And you can see very few MUC5AC positive cells. With IL-13, as is previously reported in lots of publications, increased number of MUC5AC positive goblet cells, even as early as 6 days after IL-13 exposure, and that's quantified here. So what happens then to ionocyte population as we increase the number of goblet cells with IL-13 exposure? So Guillermo developed a flow-based assay where he used different markers to identify the different cell types in the airway epithelia. And so he used tubulin, MUC5AC, NGFR, and Barden staining to identify ciliated cells, goblet cells, basal cells, and ionocytes. And you can see we can very clearly distinguish between these populations. Not surprisingly, when we treat with IL-13, you see this large increase in MUC5AC positive cells or goblet cells. But what was more surprising was that if you look at the number of ionocytes present under control conditions, and you compare that to what happens after IL-13 treatment, there's a significant decrease in the number of ionocytes. We quantified this. So here on the y-axis, I'm showing you log 2 cells per centimeter squared, control conditions in the orange circle, IL-13 in the blue, the different cell types along the x-axis. Ciliated cells decreased. Again, as expected, the number of goblet cells increased. But we're sort of surprised to see that the number of ionocytes decreased in every culture we studied with long-term exposure to IL-13. And I'm not showing you the electrophysiological studies, but there was significantly increased chloride secretion with IL-13 treatment. What we next looked at was CFTR and BSND mRNA, and with IL-13 treatment, we saw a rather significant increase in message level for CFTR, but a decrease in BSD mRNA levels. So these data suggest that IL-13 increases CFTR in secretory cells while simultaneously decreasing the absorptive function of ionocytes. So we tested the hypothesis that secretory cells are driving this increase in CFTR with IL-13 treatment. So we looked at the different cell populations and co-stained them also for CFTR expression. So here I'm showing an example from control cultures, IL-13 cultures, CFTR in yellow, ionocytes stained in pink, goblet cells in MUC5AC, more goblet cells in the presence of IL-13 with CFTR staining. We saw a slight increase in the number of cells, total number of cells over time with IL-13, and this was accompanied with increased number of cells staining positive for CFTR. Similarly, we can show the data like that. So about 20% of cells were positive for CFTR. If we look at goblet cells, really this robust increase in the number of MUC5AC positive goblet cells with IL-13, and essentially all of those goblet cells had increased expression of CFTR. Again, 100% of the goblet cells were expressing CFTR. What about ionocytes? And this is where we sort of had a surprising finding. So here we're staining ciliated cells with tubulin, again, ionocytes in pink with Barden and CFTR, and you can see, as John showed very nicely, ionocytes have this apical cap of CFTR on the top. If you do it in cross-section, you can see it there. Interestingly, when we did the same type of imaging with ionocytes, we found that while these cells were still positive for Barden along the basolateral surface, they lost CFTR expression on this apical cap. We quantified that, so again, the number of Barden positive ionocytes went down over time, and this was associated with a decrease in the CFTR positive number of ionocytes as well. And so, there were some ionocytes left after 12 days of IL-13 treatment, but very few of them expressed CFTR. I'm going to show you a couple sample images here. So here's control staining for ionocytes and CFTR. Again, nice apical CFTR caps in all 4 ionocytes that I'm showing. But even as early as 6 days after IL-13 treatment, you can see a number of these ionocytes begin to leave the CFTR cap. This one appears to be diminishing there. And then when you extend it out to 12 days, you can see that the remaining ionocytes that are present in the culture all lack this CFTR cap, while the accompanying secretory cells have rather significant amounts of CFTR expressed. So our hypothesis then was that IL-13 would decrease liquid absorption since we're seeing a decreased number of ionocytes. So we looked at this two ways. First, we did airway surface liquid quantification. And as expected, if you treat these cultures with IL-13, you see a significant increase in airway surface liquid volume. And we also did a liquid flux assay, where essentially you put a known volume of liquid on the apical surface of the airway cultures and then see how much you recover after 4 hours. And so here on the y-axis, I'm showing you fluid flux in microliters per hour per centimeter squared. And on the x-axis, time with IL-13 treatment. So a more negative number would suggest that the airway epithelia are absorbing. A more positive number suggests that they're secreting. If you look at control conditions, you can see that all of the cultures have an absorptive phenotype, which is, we know, what happens under control conditions. But with increasing exposure to IL-13, the airway epithelial cells become less absorptive. And out at 21 days, you can actually see several of the filters actually had a secretory phenotype, meaning that we actually collected more fluid or more liquid from the airway surface than what we actually put on. So there's data suggested that IL-13 decreases the number of ionocytes and decreases CFTR expression by ionocytes. And so we wanted to look at whether or not this may be the same case in human disease. So our hypothesis was ionocytes in human disease with elevated IL-13 would also exhibit altered CFTR detection. We used two approaches for this. The first, we took tissue sections from control lungs as well as humans with COPD and asthma. And then secondly, we went back to the literature and took advantage of a single-cell RNA-seq data set from lower airway biopsies in asthmatics and control and reanalyze that data. So first, the immunohistochemistry. So here I'm showing you a couple examples from a non-diseased human lung, tubulin stain in all the ciliated cells, very few number of Barton-positive ionocytes, and in a scattered number of goblet cells. If we look at it under asthmatic conditions, increased number of goblet cells as evidenced by MUC5AC positive staining, and similar thing in the COPD airway. When we more specifically look at what happens to CFTR detection on these Barton-positive ionocytes, again, non-diseased lung samples, we see this nice apical cap. But when we looked at both asthmatics as well as COPD samples, you can see, again, we retain this nice basolateral Barton staining, but not much on the apical surface for CFTR, and similarly when we looked at COPD. So our second approach was to go back to this single-cell data set and reanalyze it with the specific question of looking at what happens to CFTR expression on ionocytes. So in this series, there were 6 control cases, 6 asthmatic cases, and in total, 133 ionocytes detected or studied from all of these biopsies. So if we first look at sort of the baseline characteristics, again, more goblet cells in asthmatics, a trend toward less ciliated cells in asthmatic samples, and maybe a slight trend toward fewer ionocytes in the samples from asthmatics. What was interesting is we looked at mean CFTR expression per cell. So that's what we're showing here on the y-axis, and I'm showing you data from all 4 control subjects, and each circle represents a different ionocyte from that subject. And so we're looking at mean CFTR expression levels per ionocyte, and you can see, generally speaking, in the control subjects, we see lots of CFTR expressed on the ionocyte surface. We can quantify this, and we're 75% plus in all the samples. We saw something different when we looked at CFTR expression in the ionocytes from the asthmatics. We actually saw 2 populations instead of 1. So here you can see there's a bunch of ionocytes in the asthmatic samples that express reasonable amounts of CFTR. However, there's a second population of ionocytes that all of the donors have that essentially have no CFTR expression within their cells. And we've quantified that there, and you can look at the number of ionocytes that are CFTR positive significantly reduced in asthmatics. The last thing we wanted to look at was whether or not the number of CFTR positive ionocytes correlated with some type of disease activity. And so the authors in this manuscript also provided pulmonary function tests. And so we looked at the percentage of ionocytes that were CFTR positive in each cell versus FEV1, a measure of airflow obstruction. A higher number means better lung function. So for this control subject, 100% of their ionocytes were CFTR positive. and has very good lung function. And we saw a very similar pattern for the 4 control subjects. What was interesting is if you looked at the asthmatic subjects, we found that as the percentage of ionocytes that were CFTR positive, we saw a decrease or worsening of airflow obstruction, suggesting some relationship between disease activity. So Barton-positive ionocytes lack detectable CFTR in asthmatic and COPD lung samples. Percentage of ionocytes with CFTR was reduced in asthmatic samples, and CFTR-positive ionocyte number fell with worsening airflow obstruction. So these data suggested in asthma and presumably in other IL-13-related diseases, the absorptive properties of ionocytes involving CFTR and Barton chloride channels could be affected, while the total number of ionocytes, at least in these data, appeared to be unchanged. So I'm going to come back to my take-home points. Focused on here on CFTR-rich ionocytes and how they're important in liquid absorption. Secondly, which I found fascinating, that IL-13 in the same airway epithelia can decrease the number of CFTR-positive ionocytes, but increase the number of CFTR secretory cells. In asthma, we saw that CFTR-positive ionocyte numbers fall with worsening airflow obstruction. And again, the most important point is I think we still have a lot to learn about regulation and control of ionocyte biology. I'd like to thank everybody who contributed to this, and I'll end there.
Thanks so much for great talk.
Yeah, David, so great talk. So, in adults with asthma, presumably a major source of IL-13 is going to be memory CD4 cells, which will be in the large airway. But during fetal development, Rick Loxley has shown that the dominant IL-13 producing cell, the ILC2s, actually, see the lung during embryonic development, and they specifically park themselves by the terminal bronchiole. And he was actually making an argument that may be a way for the infant lung to regulate VQ matching when there's an infection, by turning on mucus and shutting off ventilation to an area that's infected, right? So how does this relate to early onset disease in the infant lung, and where potentially IL-13 is going to be mostly confined to that terminal bronchial space?
Yeah, so the easy answer is I don't know. These biopsies were obtained, as you pointed out, adults, late disease or later disease, and were from, I think, 3rd to 6th generations, so still pretty proximal. Also, as John alluded to, it's very striking. The number of ionocytes goes to almost 0 the further you move out into the airway tree. And so whether what I'm showing here is applicable in distal airway segments, whether it's important in early airway disease, if that's what it starts, I guess is less clear. And then this is a correlation, right? Cause and effect. I can't sort of speak to that. Hey.
Thank you so much. So I have a question about kind of speculating about a potential mechanism as to how these ionocytes are lost. And I'm wondering if you've looked at potentially the expansion of the tuft-like cells or the solitary chemosensory cells, which are potentially within the same lineage. And I'm wondering if, because those are known to be potently expanded by IL-13, if maybe more cells from this rare lineage are getting diverted into the tuft cells at the expense of the ionocytes. And then I'm wondering if maybe these sort of frustrated or incompletely differentiated ionocytes that you're seeing could just need a little ASCL3 or something like that to kind of help them along this pathway as a potential rescue mechanism.
Yeah, so we did not look at tuft cells in this data. I would imagine somebody has looked at IL-13 on airway epithelial cells and tuft cell numbers, and it seems like you're alluding to maybe it goes up.
It's pretty robust in the nose. I don't know how well that works low.
Yeah, so that'd be interesting to look at. The other problem with sort of studying the ionocytes is the numbers are already rare, and now we give IL-13, they're even rarer. So to sort of lineage trace and kind of figure out where they came from is difficult. The other sort of thing you alluded to is, could other ionocyte markers and properties be intact? So here I'm defining an ionocyte by Barton positive staining. We've tried to look at whether other typical ionocyte markers are still there, and at least in the human single cell data, it appears that they are, but again, the numbers are small.
Thank you.
Marguerite?
Hi, David.
Hi.
Very nice work. Have you ever thought about looking into other ion channels, like an octamine one, for instance, which is usually highly expressed under these asthma IL-13 conditions and IL-4 or ENaC?
You know, I'm trying to think back. So we have not looked specifically at expression. You know, there's data, I believe, that ENaC is altered with IL-13 exposure as you're sort of altering secretory versus absorptive phenotype. But we haven't looked at any specifically, either sort of immunologically or functionally.
Right.
But it would be reasonable to do.
Interesting, I would think.
Great talk. Thank you. I have a question regarding this very dramatic split of CFTR expression that you get in asthma. Of course, asthma is a very complex disease, but given the role of IL-13 that you showed, I wonder whether the ones that are CFTR negative correspond to allergic asthmatics, sort of the eosinophilic type of asthma. So is there any clinical history attached to those samples that you could use to tease apart what's going on?
Yeah, I'm trying to think. I mean, there wasn't much sort of clinical, you know, we extracted the pulmonary function test because that was helpful. I should go back and look. They had a supplemental data set where they listed the PFTs, and off the top of my head, I can't remember what else was there, but that would be a reasonable thing to, and again, it's 6 subjects.
Yeah.
We're limited by that power.
Right, but it's so dramatic.
It is dramatic, correct, yes. Thank you.
Hello. My question is, maybe I have missed some data, but does ionocyte without CFTR expression express RNA, mRNA of CFTR? My question is whether processing of CFTR protein itself is disturbed or mRNA expression itself.
Sure. So when we look at bulk with IL-13, CFTR expression, you know, if we look at the whole airway epithelia, CFTR expression goes up. I think that's predominantly coming from the secretory cells. When we stain for protein, it's not there. The single cell data would suggest that transcripts are down. And so it's not only just a processing abnormality. And we also, when we did immunostaining, we don't see any intracellular CFTR for what that's worth.
Thank you.
Thanks.
So your data on asthma and COPD is quite intriguing and very interesting. Interesting from this point of view that how could this field then learn from CF? For example, if in these people where you have found reduced number of ionocytes, if you just measure the CFTR function, I know it will not fall in the range of CF. If it is 25% of wild type, can you then give the modulators and help those people in which the CFTR genotype has not been mapped and we do not know what the genotype is, but just measuring the function, because this is such a quantifiable measure, that you find 25%, you put modulators on top of it. If those people can reach to 75 or 80%, are you going to get rid of the underlying pathology for these people?
Sure. So I think your question's independent of these findings of CFTR and ionocytes, if I understand correctly.
But did you not show that the number of ionocytes were low in people with asthma?
Correct.
And people with COPD? If the number of ionocytes are low, is it not that the CFTR will also go low?
I think it will. I guess my argument would be whether or not any of the modulators would improve that.
So,
thank you. I guess they theoretically
Yeah.
could.
Thank you.
But that would be, I think, my
I will
main
ask you
thing.
to come to the poster 264.
I will.
Anna, my graduate student.
Kevin.
David, just a minor question in the IL-13 cells that don't have CFTR at the apical membrane anymore. Is the apical membrane still connected to the lumen?
I'm trying to think what data we have to address that specifically. You know, for the ALI cultures, I think visually it is, but we'd have to think about specifically testing that question. Some of the human lung samples, we did serially sectioning a little bit to try and get at your point, because some of the ionocytes in the human lung sections actually look, they may be a little bit below the apical surface. When we serially stained them, we convinced ourselves that the Barton staining went to sort of the apical space, but I think that's a fair question, and I'll have to think about that more.
I have actually one last question. So if ionocytes' primary function is fluid absorption, and then CFTR expressing the ionocytes and also ionocyte number that decrease in the asthma, so then that creates kind of like secretory mode in the airways. And then in one aspect, it might be helpful to clear mucus if it's more secretory. But do you think that's like a compensatory kind of findings, or there is some like link, reduce like more secretion, even like fluid secretion worse asthma phenotypes?
Well, I mean, I think it gets a little at this idea of, if you make more mucus, do you need more fluid?
Yeah.
And are sort of the cells smart enough to do that hand in hand? It's possible that the CFTR you're expressing in the secretory cell is sufficient to help with that process. But yeah.
Yeah. Thank you so much for the great talk.
All right. Last but not least, we have Dr. Juan Ioanowski at the University of Saskatchewan. All yours.
Thank you very much. It's a great honor to be here. Thank you for the invitation. I'm here because my data contradicts. It's not compatible with what we just saw. We should predict from the data we just saw is that ionocytes are transporting sodium. They don't. What I'm going to show is data my students and I collected over the last couple of years using a different strategy, measuring at a single cell level in intact epithelial tissue and cell cultures. And what we are forced to conclude is that ionocytes specialize in bicarbonate secretion, are not involved in chloride and sodium transport, and club cells do most of the sodium chloride transport. Before I move on, I want to let everybody know that Saskatchewan is in Canada. If you put the finger in the middle of a map, that's what Saskatchewan is. A map of Canada, of course, not US. And my university is part of the Treaty 6 territory, the homeland of the Métis. I did not know what the speakers will mention before me, so I made a very, very spooky and flimsy introduction to adapt. So I'm making as I go. What we all agree, I think, is that the airway epithelium is very complex. It has at least 11 different cell types, and they organize, we see them here in this Montoros figure, and they organize into a sheet of pseudostratified epithelia, and all of these cells together work as a team to produce airway fluid that has to have the right osmotic concentration, the right concentration of antimicrobial properties, antimicrobial compounds, the right type of mucins with the right viscosity. The discovery of ionocytes has called the attention of all of us, and there has been a huge attempt for many, many labs to try to figure out what they do and who does what. So far, what we've seen today, and is the most preferred strategy, is to create genetic models that allows the researchers to shut down one cell at a time. So the exercise allows for results like this. The paper by LL05 from Jan Thorner's lab and Feng Yuan from John Engelhardt's lab both conclude using mutated cells that ionocytes are moving sodium. You can see sodium transport through ionocytes according to the paper by Ley, and secretion or absorption of sodium and chloride through ionocytes in ferret tissue. This is, as you saw, extremely convincing data, especially the data were reintroducing CFTR in a CF background produces significant changes that make sense. So the prediction from this is that if I were to measure a transport across a ionocyte, I should see sodium transport. I'm going to make a little side note here and try to explain how I see epithelial transport across this epithelium. Epithelial transport depends on the work of all of the cells. So if a muscle cell contracts and a neuron transmits electrical signals, an epithelial tissue bends the laws of thermodynamics by creating localized electrochemical potentials that move ions in a direction that otherwise would not move. So the transepithelial potential, the paracellular pathways, the ability of solutes, water, current to move in between cells paracellularly, they're all determined by the entire epithelium, whether the cell is transporting or not. That means that whether there is electrical coupling between apical and basolateral membranes is a feature of the entirety of the epithelium. So it's a team sport. So a very unfair and crude analogy of using knockouts to understand this will be having a soccer team, originally from Argentina, so all the sports I talk about is Argentina, in the soccer, grabbing a soccer team, deleting their goalie, concluding they don't score anymore, and the conclusion we do is the goalie is supposed to score goals. That's a risk. Of course, this is very crude and unfair, the controls they did, it's not, but I propose in 2023, when I saw these papers published, to use a different approach. Instead of mutating the tissue, work with a normal tissue and identify at individual level what each cell is doing. This is not the first time this has been done in the field of science. We have here in this room Kevin Foskett, who invented ionocytes. He did in the 80s a lot of research doing single cell measurements of ionocyte function in fish gills, and he used a technique called current density probe. And the idea was you could measure at a single cell level what every cell in the epithelium could do. And he described magnificently how, together with Terry Macken and Dr. Shafi, how they worked. We use a technique that is an evolution from his technique. Instead of using voltage sensing electrodes, we use ion selective electrodes. And the idea is extremely simple. In this cartoon, I'm going to try to explain it. It's very, very straightforward, so there's no effort into this. Here we represent an epithelium. This is the lumen of the epithelium. This is the blood side, basement membrane, and these are all the cells in a pseudostratified epithelium. This arrow here represents a hypothetical transport by a cell, and in this case is moving sodium from the lumen to the bath. If there is no one mixing the fluid with a spoon, so there is no movement, it develops an unstirred layer around the epithelium. And in that layer, what happens is the cells start depleting the ion that is transporting from the liquid adjacent to the cell membrane. And it creates a gradient of concentration of ions and any other thing that is transporting. So one can measure this gradient and calculate how fast the cells are transporting. So that's what we do. We put a sodium, in this case, a sodium sensing electrode close to the epithelial cell that we're moving away. We measure it again, and we can calculate how big is this gradient, and there is a mathematical approach that allows us to determine how fast the cell is transporting. So we can grab this setup and we can put it on top of the ionocyte, and we can measure at a single cell level with micrometer resolution. So we can tell this cell apart from the neighboring cell. We can do this with any cell we want. This is called ion selective self-referencing ion electrode technique. The expectation from the data that has been published by John Engelhardt and Ian Thornell and others is that if we put the system on top of this ionocyte, we should see a sodium gradient when we stimulate with 429 BMX. So we tested it with initially human bronchial epithelial cells from primary cell culture from non-CF and CF tissue, and then what we saw was no sodium transport. So up here we have the result of a sodium sensing electrode. So this is the flux in picomole per centimeter square per second over time in seconds. And we see that there is no sodium flux. We put fractional IBMX and we see nothing. But when we use a proton electrode, so a pH electrode, or a chloride sensing electrode, we do see a deflection in response of fractional IBMX. So the cells are producing a reabsorption of chloride, so chloride is being moved out of the lumen into the blood side. So this reabsorbing chloride, and simultaneously we see that protons are disappearing from the basolateral side. So the lumen is becoming alkaline. All of those transports are lost in CF and they're recovered when we treat with ETI. The issue with this is that, as I mentioned before, the properties of ion transport depend on the neighboring cells. And it's entirely possible that our HBE cells did not produce the proper environment for ionocytes. Therefore, what we decided to do is to directly work with tissue. So we started working with swine distal airway. And the idea is if we take the airway out, we do not culture anything, we just take it, kill the pig. Within minutes, we take the tissue and we start measuring sodium, proton, and chloride. And what we saw should represent what should happen in a normal tissue. Again, we saw no sodium transport, but we saw a chloride reabsorption and alkalinization of the lumen. Next, we thought the problem is that we are using distal airway, and the work published by Feng and Lei were upper airway, were trachea. So we started to work with trachea from the same pigs. So this is a cartoon from one of John Engelhardt's papers. We can measure in the surface epithelia up here, but we can also measure in the distal part of the gland. Both of them has ionocytes. And we measured the same ions, we saw the same thing. No sodium transport, only an alkalinization of the lumen, both in the surface and in the distal part of the gland. So again, this is alkalinization of the lumen of the mucous tubules. It is problematic. I don't know why we don't see the same thing that my colleagues publish. I have been discussing with Feng, why am I wrong? We still don't know. So hopefully I'm going to start working with different models to try to figure this out. But in our hands, I must conclude that ionocytes do not transport sodium. They only produce an alkalinization of the lumen. And this alkalinization of the lumen is the result of bicarbonate transport. This is something we published a year ago. But we repeated it in pig tissue. If we block CFTR, or if we remove all the bicarbonate and block the carbonic anhydrous acetosolamide, we get that the effect of fractional IBMX is abolished. So in black here, we see the effect of the drugs, and in red, the control. So it is moving bicarbonate. And the movement of bicarbonate we propose is through a chloride bicarbonate exchanger. We propose that because that exchanger was found with, we found it using antibodies, and also it's been shown to be, the mRNA shown to be there. So when we block with the IDS, the exchanger, we have no transport. So in our hands, I'm sad to say, ionocytes only transport bicarbonate. The model that we propose is essentially a carbon copy of the model for pancreatic duct cells bicarbonate transport. It is a AE2, so a sodium chloride bicarbonate exchanger that works in coordination with CFTR. Bicarbonate is pushed out in exchange for chloride, and the chloride is replenished through CFTR. This is exactly the same as was proposed by Novak and Greger in 1988 for the pancreatic duct cells. You can see this exact same thing, the bicarbonate chloride exchanger in conjunction with a chloride channel. That makes me a little bit happy because this model has been tested forever and it still remains valid. However, we do measure sodium and chloride transport, but not through ionocytes. When we measure across club cells, not ionocytes, but club cells, we find both a sodium and a chloride signal that indicates that when stimulated with phosphorylated IBMX, club cells move sodium and chloride into the lumen. And there is only a very small bicarbonate contribution. And this is lost in CF and recovered with ETI. In the distal airway of pigs, we get the same thing. So it's not an issue of anatomical distribution or a particular arrangement or some issue related with the cell culture. The tissue does the same thing. So we had to conclude that from our data, in the epithelial tissues that we studied, there are two different functions for ionocytes and club cells. The ionocytes in our hands are bicarbonate secreting machines, and the club cells produce most of the volume of the ASL. We wanted to see if we could separate these two functions, and we decided to take advantage of one of the descriptions of Dr. Kenichi Okuda in a 2021 paper in the Blue Journal, where he showed that UTP does not stimulate ionocytes because ionocytes don't express the P2Y receptor that is required for responding to UTP, but club cells do, and they showed the transport by secretory cells. So we tested that, and yeah, indeed, UTP does not stimulate ionocytes, but it stimulates it stimulates club cells. So it should be possible to distinguish between a liquid produced by the same epithelial when stimulated with UTP, which only contributes club cells, or phosphorylated IBMX, in which both produce fluid or produce transport. We can also block this transporter with the IDS. It's another approach. And blocking with the IDS should also separate between the effect of ionocytes and club cells. And our hypothesis was, based on literature, that bicarbonate secretion may be important for the unpacking of mucins. So we decided to study the viscosity of the airway liquid fluid using FRAP, fluorescent recovery after photobleaching. So we collect fluid, either stimulated with phosphorylated IBMX or UTP, and with or without the IDS, and then we photobleach and see how fast the FITC label dextrans return to the photobleach area. So we got this cartoon just shows that. There's a photobleach here, then it recovers, and then half the time it takes to recover 100%. We plot that as a measurement, an estimation of viscosity. You see with phosphorylated IBMX, it takes about 5 seconds to recover. If we put the IDS, it takes longer, about 7 seconds. If we put the IDS, but we include external bicarbonate, it recovers back to 5 seconds, and UTP makes it quite viscous. So with this data, we believe that we are showing that the ionocyte is producing bicarbonate, and the bicarbonate plays a role in determining the viscosity of the ASL. So just to finish, again, I agree with my previous speakers. I do not know which cells should be targeted, but our data seems to indicate that both cells play a role, and the roles are different. The ionocytes secrete bicarbonate, maybe for mucin unpacking, and club cells produce most of the ASL. Barring the conclusion from Dr. Engelhardt that we can target other cells, a strategy that only targets ionocytes will produce a dehydrated fluid, and if you only target club cells, you will have a viscous fluid. So with that, I would just want to mention my students sitting at that table, Jay, Amirala, and Sarah. They did most of the work, and you can see their work in poster 156 and 189. And Amirala's work on submucosal glands is going to be presenting the talk on Saturday, the 25th. And finally, my collaborators, Veronica Campanucci and Julian Tam, and the funding agencies. Thank you very much.
Go for it, John.
Great talk. Is it on? Yeah. So your ion probe suggests that above ionocytes, you're absorbing HCl. Is that right?
Correct.
You said protons and
Yeah.
So, and the result would be alkalinization, right?
Yes.
Because you're taking HCl out.
Yep.
You probably drive bicarbonate going out. Do you think that can drive fluid absorption? I mean, plus the channel CFTR it's got chloride going out rather than in, which I think your point is it only goes in.
So I'm going to answer your question with this cell, which is much better established than the ionocytes. In the pancreatic duct cells, there is also a net proton reabsorption. So you lose the bicarbonate in this direction from the carbonic acid, and this proton needs to go away. Now, this proton cannot just live forever as a proton; it must be buffered. So what we think is happening is that this is something I discussed with Ian before. The proton that we are absorbing here is probably buffered; otherwise, you're going to get the wrong pH. And then the chloride becomes an orphan anion that's going to require a counter ion. So it is possible that there is another pathway of sodium reabsorption that can work together with ionocytes. So our results are much less incongruent with your data than Ian's. Ian, I can come up with a way in which this results in reabsorption, but I do not have. We measure about 100 ionocytes. We've never seen sodium transport. One of the problems. Sorry.
And I'll just clarify. I mean, we have not. The only reason that I show ENaC within an ionocyte is based on the single cell mRNA experiment. Yeah. We've never done a biological experiment that said sodium is going through the ionocyte. So, I'd say that we're not as in disagreement as possible. I mean, the biggest disagreement, I think, in the field is, do ionocytes contribute to chloride secretion and water movement, so salt movement into the airway surface fluid?
We don't see that. We never see that, but so one of the advantages you have with the model is that for us we need to use pharmacology, and chloride transported pharmacology sucks, in essence. And they are very bad. All of them are very bad blockers. Everything is interacts with something else, and every time you want to block something, you block someone else. And also have a lot of different cells that all of them want to bother me. And no one wants to do exactly what I want them to do. So, and that's our main problem when we try to publish, is that how do you know that you're not just, so that disadvantage that we have is that our model is very, naughty. It doesn't behave, it's like my daughter.
Plus I'd say that the other big thing when you think about electrophysiologically comparing both the experiments with ours is we add DIDs before we either make a gradient or we activate CFTR. Now, in the absence of a gradient, you still need ionocytes to secrete chloride. At least that's the data that we.
So I've been talking with Feng about trying to use our technique on your cells and see if we can figure this out, because I think your data showing that maybe we don't need to target ionocytes is I think it's inspiring. It's a good, if it's true, because with 20% efficiency of liquid nanoparticles delivering the genetic material to the basal cells, you're going to end up having, if it's just by random, you're going to end up having 0.09% of ionocytes being transformed. So it would be great if we don't need to get the ionocytes.
I agree. You ought to take the GFP knock-in and put it on here and use the conditions both in the short circuit current that we measure with DIDs before you do your measurement to try to get some congruency with why.
This has to make sense at one point in our lives.
Great.
Hey, nice talk, Juan. Beautiful work. I'm going to have the same disclaimer as John, that we stuck ENaC in that cell based on single cell RNA sequencing. We put that in the legend, and I think your data is pretty convincing that cell is not doing that. So I guess my follow-up question to that is, where is that cell?
So I'm gonna say something that I shouldn't. We sent a paper for review, came back rejected, but one of the comments was excellent. And it was a comment that I think we had this discussion before too. Ciliary cells also transport with this single cell. So we do 2 ciliary cells that responded and seems to be CFTR mediated. 2 out of.
For ENaC?
We only did CFTR stimulation.
I see.
But my point being, maybe that's the path.
Yeah.
And it wouldn't have to be that big of a path because you have a massive number of cells. Small transport will be fine.
Yep.
So we don't need to counter ions for this guy. The transport is, the ionocytes that Kevin Foskett showed in fish gills, they're like cannons. They like the amount of current they move is like crazy. These guys are not like that. They're pretty much, it's a lot more calm. So we don't need a lot of sodium pathways and sodium transport to provide the counter ion for this stuff. So it could be CLL cells, and it would also be congruent with what Dr. Engelhardt was saying.
Yeah.
So we're gonna try. Also, we're talking with Feng that what we need to do is instead of now what we do is we try to find a ionocyte and measure a ionocyte, and then try to find a club cell and measure the ionocyte club cell. What Jay has decided to do is to, instead of doing that, to do a survey essentially. So grab an area, a random area of the tissue, and our computer can do a grid scan. We can just program the computer to scan every 10 micrometers, and then we can do a grid scan and then overlay that with immunological data showing which cells, the identity of every cell. So we can overlay the two, and then we can have a better understanding of what other cells are doing. So that's what we're trying to do. Now we're trying to do also the other thing. I think what we need to do is to work with human tissue. So I've been trying to get a human transplant. My city, Saskatchewan, in Saskatoon. It's a great place, but it's tiny. It's about 300,000 people, and we'll have a lung transplant program. Our patients go to Alberta, which is the neighboring city. So for me to get tissue is hard. So I am trying to create collaboration with Montreal, with Toronto, with Vancouver. But it's a huge problem. But I think asking the same question in human will be super important. And COPD, the COPD question, I think is also super interesting, trying to see if these cells do different things. So that's the path that we're trying to go.
Yeah. I mean, I think it's a lesson for us all here that single cell RNA sequencing can only take us so far. And at the end of the day, we have to set up experiments and look at the physiology.
Yeah, I want a disclaimer. We published a paper this year with the CFTR knockout pigs. So I do use knockouts. I do use genetically modified animals. So I'm saying I use them too. So I'm sorry, I didn't mean to come across as an asshole. Hi.
Hey, Juan.
How are you?
Yeah, very, very nice talk. I really appreciate your football analogy because I am from Peru.
Yeah, I know.
The IDS is a very non-selective inhibitor of an ion exchanger. So do you have an orthogonal means of proving that is the chloride bicarbonate exchanger, what you're actually silencing?
Well, so we don't have it and we never will, because that means being a century of trying to block chloride transporters, and they all suck. There is no. So the only thing we can do is try to use genetic models, which was one of the comments from another reviewer that rejected the paper, to use genetic. So that is probably the way to do it, to combine this untouched natural thing with very advanced genetic models like John uses. But being specific with pharmacology for chloride transporters is a lost battle.
Yeah.
You're never gonna.
So I guess it's more of a phenotypic observation rather than isolating an individual molecular entity.
I can hand wave my way through this and try to convince you, but the truth is the idea is it's not a specific blocker. It is not.
Thank you.
Yeah.
Hi, I'm also from South America, so I love the analogy. So I mean, all these talks, what they made me talk is that it seems like it's a very complex and dynamic environment in the airway. So I was thinking whether it's possible to do these experiments with like mixed cultures where like only one cell type at a time will express CFTR and see whether other cell types will compensate when, for example, the ionocytes do not express it.
Yes, you could. It will be my cup of tea, because there is a very close relationship between the anatomy and the function in epithelial tissues, right? So the kidney is all anatomy, and the cells are in one part of the kidney, and not if you move it anywhere else, it just doesn't do the same thing. Ionocytes in the kidney have the intercalated cells can be A or B. The A produces acid, the B produces base. It's very specific. So I find value in working with the complex native tissue. That's what we are good at. I'm never gonna compete with John or with David, and they're gonna eat me alive. What I can do well is work with complex tissues right out of the lung. It can be done, but the result is gonna be super difficult in my mind, to evaluate, I think.
Also, whether you have explored in the context of like an inflammatory environment, if the cells change their chloride transport.
Yeah, so that absolutely is, the asthma and COPD, that is absolutely very good. And now, what we are doing now is we're grabbing, one of the things that is happening here, we're using Forsonic PMX, all of us. But that's not really a physiological signal. So we are trying to find what are the physiological signals that control the function of ionocytes and secretory cells. And so far, forgive me, Jay, because I'm lying here, but so what we think is happening is that ionocytes are more responsive to, they respond to LPS, for example, or to cytokines, but they don't respond to VIP. So we think that it could be also separation of function. So the ionocytes might be related to the release of MAC5B and MAC5AC, but not to other types of mucins, and it could be related to responding to infection conditions, and some other combination of cells could respond to another type of insult, like inhaling ozone or something like that. So yeah, but the data for that is super new. They did it last month, and there's not enough data.
Thank you.
Last words, Dr. Foskett?
Yeah, just a couple of comments, I guess. First, if you take your ionocyte model, you can make that a sodium bicarb secreting epithelium if the paracellular junction is sodium selective, or you can make it an absorptive epithelium. And so I think what this field needs, and it's very technically difficult, is to know something about the paracellular junctions, and are they different depending upon which cell type the ionocyte is making a junction with, and what is the chloride concentration in an ionocyte? You need to know driving forces, thermodynamics, to really know whether any of this stuff works. What's the apical membrane potential? What's the basolateral membrane potential? That's not even well known for the surface epithelial cells. You have to go back to Knowles and Boucher, and that was before, some really using ion selective microelectrodes, which, you're using them beautifully, but you try to put them inside a cell.
I did my
so
PhD.
These are measurements the field is still missing, and I think maybe part of the resolution would be to develop some techniques to get into these rare cells and make those kinds of measurements.
So I did my PhD measuring ion selective microelectrodes inside the cells. I measured sodium, potassium chloride, proton, and I calculated all electric potential for the fly kidney. It's impossible to do here because the electrode tip has to be 100 times smaller than the cell. That means that we have to 0.1 micrometers. 0.1 micrometer. It's not going to work. It can only be done using dyes. But yeah, I hear you.
Great. Well, on that note, I just wanted to thank all the speakers and the discussants for this really thought-provoking session. I think we have more questions than answers, so that's the beauty of science, guys. So thanks everyone for joining us.